And first of all, I really would like to thank everybody for the fantastic organization. And my work actually today is pretty easy because yesterday you already had a very nice introduction by Professor Subramaniam on all the overview, especially on univariate statistic analysis. So I will go very fast in that part. And I will do something different because what was asked me to do it today is to try to explain biostatistical approaches more in visualization and interpretation. So it is Friday, it is day 5, and so I'm going to not add more math. There is always time to go through the details on the methods, but it is better to see also for beginners which is the feeling using the different methods. That is my aim of today. So I think I have to reduce this. Okay, so due to the short time, I will simply mention the statistical hypothesis testing that were explained yesterday, to go then to how regression methods work, and to focus the most part of this presentation on multivariate analysis. But before deciding which is the method, the biostatistical method we want to apply, you should ask yourself, how do your lipidomics data look like? Indeed, we have different data matrix. We can have data matrix like this, where we have our samples, experimental sample measured on the row, and our variables as a column, with no response variables. Just this, we want to interpret what is in our data. A second possibility is the same X matrix, but we want to correlate it with something, with another variable. Or we can have more than one variable to be correlated to our X matrix. And this is, for example, very common in clinical approaches where you have maybe a lot of clinical parameters. You can have even more complex structures like three-dimensional matrices, but for today, and also due to the limited time, I will stay in these few examples. If we go more into lipidomics application, especially in untargeted lipidomics, sometimes we're going to have, quite often actually, we are going to have a quite fat matrix because we have a limited number of samples with respect to the number of variables of lipids that we are able to identify so far. And we can have multiple response. What is important to underline here is that our X matrix is characterized usually by collinearity. That means that our variables are often not independent, because this is the nature of lipidomics, right? And also Y could be not independent. In addition, these are our X matrix is made by using experimental values, so we can have also noise in our data. So when we are going to apply statistical hypothesis testing, they are pretty useful, especially if you know what you want to check. For example, you are making an experiment where you have two populations of samples, you have your controls, your treated samples, and you want to compare to see if they are statistically different. And yesterday you had the proof on how it is important to remember that many of these methods are based on the assumption that the distribution of each group can be defined as a normal distribution. Otherwise, there are several other ways to work. And yesterday you had a lot of information about that. Going to the visualization of this data, that is my aim today. Like Valerie was mentioning in the first day, volcano plots are pretty well used. And basically they are combining the information coming from the statistical significance in terms of p-value and the fold change variation. While in terms of p-values, usually in the papers you find or 0.0 one value to say that it is highly significant, or a value that is less than 0.05 to say that are significant. But like Professor Subramanian was saying yesterday, for fold change analysis, this variation is much larger. It depends on the nature of your data. Basically, when you are reading a plot like this, you have to consider that you are fixing some thresholds like here and like here, so fold change, and a threshold for log p, and therefore you are interested usually to interpret what is happening in these two areas of the plot, because these are the variables that are passing both filters. Again, as Valerie was mentioning, these points, it depends on the data you are plotting. These points could be not a single lipid, for example, because they are coeluting maybe somehow. But you can also have that you are plotting the mass to charge at a certain retention time before doing any adduct clustering and so on. So the same lipid species can be, this is a real example I was looking for, the same lipid in different adducts. You see, they will not collapse in a single point. So if the question is, what is better to do? When is better to do the volcano plot? Remember that the volcano plot is a tool to plot your data. So it depends on the question you want to answer yourself. Because even here, you have an information. If you have the same trend for multiple adducts, you can have an additional information that maybe can be of interest for you. Another approach that sometimes people are doing is to coloring the volcano plot by the lipid subclasses, for example, after an identification. In this way, you immediately will see if a certain class, for example, here, this green, dark green class, is always present in the same area. So that could be an additional information. So you can explore this volcano plot in several ways. Let's move now on regression methods. We are all very familiar with linear regression analysis. It is very simple to do. And just to remember, linear regression analysis allows you to see how one variable can be used to predict another variable. And so the straight line equation is the basis of this regression, okay? Which is the driving modeling? The driving modeling is the minimum sum of square difference between the experimental value and the true value. But here, again, I'm interested in reading the information from the plots. And look at here, you will see that I am projecting my experimental points in a fashion that is parallel to the y. This means, from the statistical perspective, that I am assuming that the error is only on the y, not in the x. Otherwise, I could use other methods to project them, okay? So in linear regression analysis, I am assuming that the error is only in the y. Another important point that we have to take into account when we use regression, linear regression, for example, is that correlation is not causation. People that are familiar with statistics have very well clear these points, but let me make a couple of examples for people that are at the initial state of their studies. One of the most used examples to say that is the correlation between the number of stork pairs and the newborns, okay? There are papers that are showing data for which we can have a good correlation, okay? Here I'm giving you several literature if you want to go deeper into that. But that doesn't mean, at least we don't have the proof to say that storks deliver newborns. This can happen if, for example, there is another variable behind that can explain this as a secondary effect. So let's make an example that is sometimes reported as an hypothesis. We can have that the measurement was made in an area that was expanding, and if the population was expanding there, we have naturally more newborns, but we have also more houses, so we could have more nests, and so we could have more number of stork pairs. This is just to give an example very simply, and you can read the papers. If you go deeper in the web about this, you will find other funny cases like the correlation of the per capita cheese consumption with the number of people who died becoming entangled in their bed sheets. Okay, so my message here is that you have to remember also here that regression is a statistical tool, not a cause and effect statement. So once you have a nice line, nice fitting, ask yourself if you agree with this interpretation. That was a simple case because it was a simple linear regression. But in the omics, we usually have X matrix. I showed that before in the first slide. And so we can apply multiple linear regression. Again, it's good. It has, all the methods have their pros and cons, but you have to into account what we have said for the simple linear regression, so that also MLR assumes errors only in the Y, okay? So we are assuming that we have already a good matrix here. And we are also assuming that there are no interactions between my variables, okay? And so we could suffer for this multicollinearity that is in the nature of lipidomics data. So these are assumptions that we have to take into account when we decide which method we want to use and which is the aim to apply this method. This is why multivariate analysis is largely used in publication. And in particular, today, since I have a short time, I prefer to focus the attention on chemometrics methods, also because in the presentation we had in these 5 days, there were a lot of PCA, and therefore we have the proof that is one of the most used methods. What is chemometrics? Chemometrics is the application of mathematical, statistical, graphical, or symbolic methods to improve the understanding of chemical information, and can be used at different times during your analysis, from the very beginning, simply to inspect the data. I will show you why. And also for making more deeper interpretation. The definition of chemometrics is pretty old, I would say now, because it is almost my age. It is 1978 by Svante Vold and Bruce Kowalski. But the explosion of the chemometric tools actually came with the widely use of personal computers and in the omics in the 2000s. And today, chemometrics is considered a subset of what you call machine learning and artificial intelligence, although actually there are aspects of chemometrics that could be out of this group. Chemometrics is widely used in lipidomics because it was developed to deal with collinearity. This is the first point. So for a chemometrics approach, collinearity is not a problem. In addition, it starts with the assumption this time that raw data are always noise, and at the beginning they can be also meaningless. So we need something to extract the information and have the interpretation. So this fits a lot with the natural complexity of lipidomics. And so now I would like to tell you a little bit more on how chemometrics works. The main research in chemometrics is what we call pattern recognition. Again, this is for a person. I would like to spend some time in that because maybe not all of you are trained in what pattern recognition is. So pattern recognition is when, for example, you have 2 or more observations that are made on each member of a set of sample, and there are patterns that can result, that can be used to classify or characterize the groups of samples. This seems a very strange definition, but this is actually how our brain works. So if you try to read these 2 sentences, I'm sure you can. We all know that our mind can read this, and the order of the letters is also not so important. Although these are terribly written, I wrote this myself, our brain is able to read it because it is using patterns. It is not important to have all the letters in place. we are extracting what it is in the text. Another example is here. Our brain in images, it's very good in understanding who is the female in this group. These are people from my group. They all wear jeans and they wear masks, but we still have the metrics to understand what is going on and who is the female. But not always the story is so simple. And for example, this is my sister child. And if I'm asking you if it is my nephew or my niece, it could be difficult. And we will start applying pattern recognition strategies. For example, the blue pacifier can say that he is a little boy, but the pink dress tells us that maybe it is a little girl. By the way, it's my nephew Filippo. And this is exactly how principal component analysis works, okay? Pattern recognition is in the basis, and it applies pattern recognition in a high dimensional space. Which is the goal of PCA? The goal of PCA is to reduce the dimensionality of my data. Remember, we have a fat matrix, big matrix, data matrix, while retaining as much as possible the variation present in the dataset, like Professor Subramanian was anticipating yesterday. And PCA is an unsupervised learning method. That means that works only with my X matrix. Some people prefer to talk about compression algorithm, but actually it is a reduction algorithm, and I'm not going into details with that. And now, instead of making the explanation on how PCA works using the matrix explanation or mathematical approaches in a linear combination of variables and so on, I prefer to use here again a graphical approach to understand what is going on. We have our X matrix for to run our PCA. We have objects and variables. That means objects are our experimental data, our samples measured, let's say, and variables are our lipids. And since our brain is limited, let's imagine that we are working only with 3 variables. This doesn't happen in reality, but let's work with 3 variables. Each object, so each experiment I'm doing, can be plotted in the space defined by x1, x2, and x3, my 3 variables, using, for example, the peak area that I have here, like a coordinate in the space, right? And I can repeat this with all my objects. And at the end, I will have something like this. And I will start looking at my data already here, and I can see that there are kind of clusters of my data. But this is something that usually I don't see at the beginning, right? So PCA is helping me to decipher what happens here and how it works. First, it starts with the most simple, the simplest, let's say, model we can apply, that is a line, right? To try to reduce the distances and to explain the maximum variance on my data. And so that would be the first line. But still, you will see that some points here and here are not well explained, right? They are too far away. So we can use a second component to try to explain this other direction of my data. And so all the points can be projected here. As you are seeing here, I am not projecting in a specific line parallel to the variables like in the regression, but I'm going orthogonal to my variables like here. And so I'm taking into account that the error is in all my variables. But 2 lines define a plane, right? So this time I can, if I'm working with 2 principal components, I can project all my object, all my experiments, run sample, in this plot. And this is how your PCA score plot is made, okay? So we will have principal component 1, principal component 2, and all my points now are projected in the plot, and I can see if there are clusters formed. And usually, if I can see clusters here, and especially if these clusters are actually what I know about your data, what I'm expecting, I will be very happy. And the next step would be why, to understand which variables are responsible for this separation. And so that is the second question. To know this, so to interpret our score plot, we need another plot, which is called the PCA loading plot. That can seem more complex, but basically we are now describing the orientation of our principal component in terms of the cosines for the variables, for each variable, okay? And so we are going to have for each principal component 3 values, the cosines of the angles alpha 1, alpha 2, and alpha 3. And these values indicate how the original variables are loaded into the PC1. This is why it is called loading plot. That could seem a little bit complex, but at the end, you have to consider that this is very useful because this time you have your variables projected exactly in the same plot defined by PC1 and PC2. And since they are in the same plot, defined by the same variables, here I'm calling PC1, T1 in the score plot and P1 in the loading plot, just to differentiate them, you can superimpose your plot, the score plot and the loading plot, because they are exactly equivalent in the space. And here you can start your interpretation. Remember that your experiments are here and your variables are here, okay? And let's imagine that we are very happy because the separation of the blue and the red points was actually our goal in our experiment. And we want to interpret what is going on here. We have to go in the second level here. And the light blue variables that are localized here have a higher value for blue points and lower value for the red points. The orange variables that are located here have a high value for red points, so our objects here, and the lower values for the blue. And all the variables that are in the middle are not important for this separation. Maybe these variables can explain something else, but if we are stopping at this level, all the variation is simply along the first principal component. So I need only to see these variables that are at the edges, okay? And so if my goal was this, I'm fine. I can work only with one principal component. But my suggestion is to go a little bit further and try to see what happens also in a second component, although what you search is here. Sometimes you can find a nice secondary effect in another principal component. It could be that this is the majority of the differences between control and treated sample, but the second component is telling you that these are the old samples and these are the newest samples collected. This is simply an example to tell you that if you are able to interpret what happens in another variable, maybe that could be a good way to add a second variable here. And so the next question would be, how can I stop? How can I know when I can stop adding a new principal component? Again, you can find several ways because there is the mathematical approach that we are going to say later based on statistics. If you are not a lot into that, usually you will find together with the score plot and the loading plot also the residual plot. And if your residual plot still has an information, you see, there are certain separation, you can decide to add another variable, another principal component. If you cannot understand what is going on, maybe it's better to stop. And this is a nice publication that I suggest you for cross-validation in PCA, for example. Here is the stopping criteria by eigenvalue that I'm not telling you today due to the time, but the best criteria, especially if you're not an expert, would be stopping by interpretation. When you are not able to interpret your data anymore, it's better to stop. Now that I have explained the PCA, partial least square regression, or PLS, will be very, very simple, because the goal of the PLS will be again to reduce the dimensionality of the data, but this time we are looking for a correlation with the Y, so with the response data matrix, okay? And so we want also not only to make the dimensionality reduction, but we want to retain the dataset variation to maximize the correlation with the Y. Since we are adding this information to the model, PLS is a supervised learning method. But at the end of the day, the interpretation is pretty similar. You have additional information to look for. Indeed, you can again have your score plot, your loading plot, you have weight plots, you have several plots in here, but in the loading plot, you will find also localized your Y. Here we have the first Y, second Y, and third Y, okay? And so from here, you will notice that one of your response variables is not important for the separation you were looking for, right? But the other two variables are nicely correlated with what you are searching. And you can also see which lipids are actually directly correlated with this Y or inversely correlated with this Y. I hope you see my mouse. I hope so.
Yes.
Okay, thank you. And the same for this other variable. We are going to have variables that are directly correlated and inversely correlated. So that is an additional information that can be used also to inspect for biomarkers, for example. In reality, unfortunately, in lipidomics, it's not so useful, not so common to have Y that are continuous values. That is what happens actually here. PLS assumes that here you have continuous values. What you usually have is the knowledge that some object belong to class 1, some other object belong to class 2, and so on. And so you can use a different kind of PLS that is called PLS discriminant analysis that will try to find the best correlation based on the classification that we have done. Please remember, in the case you are using software in which you have to type this data manually, this is the correct way to write if you have 3 groups. So you don't need to put, you don't have to put 1 and here 2 and here 3, because they will be different numbers, okay? Or making only one line like 1, 2, and 3. This is the correct way to write. In this way, the groups will have all the same weight. Let's say like this, in a very simple approach. So don't use a single response 1, 2, and 3. This is much better. And now let's use the last minutes to make some real case studies, okay? What can happen when you are applying a PCA? Let's imagine that we have 3 classes, controls, treated and untreated samples, but our method doesn't know, because if I want to apply PCA, I'm not providing this information to my method. And I can have 2 possible outcomes here. One is here. So the first 2 principal components are actually not able to separate anything. And this doesn't mean that other components are not able to do it, but there is another variation that is more, let's say, it is stronger in your data. And it could be a batch effect, or everything can happen in your data. So it can be informative anyway. Or I can have a second possible outcome, like here, where in the principal component analysis, I can see already the groups formed, what I am looking for. So the first two components are already giving me information, If I am in the second case, I can start my identification looking to my loading plot. And I can start inspecting the variables that are responsible for a high, for this separation. So here, for example, I will have lipids that are higher in controls and lower in untreated. Here I will have lipids that are higher in treated and lower in untreated. Here I will have lipids higher in untreated and lower in controls and treated. Okay? But if I am in this first outcome, I can consider to use a supervised method instead. This is not forcing the method in a general sense, because it could be that you really need to extract some information that is in other components that are in the PCA that are too far. But you will have usually lower level of separation. Here, for example, the method was able to separate one of the groups, but the other two groups are actually collapsing in these two components, okay? And now I would like to end with a few advices. The first is always control the number and the distribution of empty cells in your matrix. This method is able to handle, to deal with empty cells, missing values. Usually they can, in the method is okay if you are like 20% maybe of your missing value, but not too much. But it is good if these missing values are randomized in your matrix. Sometimes this is not the case. You can have some variables that are very low in abundance, and so in a very long runs, it can happen that the method, the analytical method is maybe suffering at a certain point, and you are losing these low values in some samples. And if it happens that the missing values are mainly in one class, that is really detrimental, because this variable will become very important in discriminating the control with the other ones. So have a look at the matrix first. Second case, I suggest to use PCA from the very beginning, simply to analyze your data. As soon as you have your data, you can upload and search for outliers, because I will show you a real example in which outlier can be hidden by other supervised methods at the end. And maybe this is really an experimental data that suffers for some instrumental problem or extraction problem. It's better to know before. And this is the advice on apply PCA before applying supervised methods. And this is an example, a real example. And here I reported also a paper that I found very nice on PCA as a practical indicator of OPLS-DA model reliability. This is my PCA. These are my real data. I have a nice separation between the green and the red already, so the signal is quite strong. I can apply PLS-DA to help me in the interpretation. And I can apply also OPLS-DA. I didn't go through that, but it is a way to make this separation more evident to help the interpretation in an orthogonal fashion. And here it's very simple. All the variables that are on the right are higher in the red and vice versa, right? But now I am applying this to this, my bad data, okay? I have an outlier here. And even if, even if I remove my outlier and I recalculate my PCA, I have this. They are mixed. But now I do in the same data from here with this big outlier, I'm doing PLS-DA. I start to see some separation. I do my OPLS and I do something magic. I have the separation on my data. This doesn't mean that this is not real, but that could be because this is the work of PLSDA and not PLS. But I am losing the information of one data that I am classifying like a pink, and it will have an impact in my pink group. So it's better to do this before. So this is the end. My take-home message for today is to be always curious when you are applying biostatistical approaches in lipidomics. Ask yourself why your model looks like that every time, and be wary of too good models because sometimes could be a problem. And if data are bad, nothing can be obtained out of them. And here there are some papers to explore more deeper what I'm saying. Thank you very much, and I'm ready for questions if you have.